Unexpected though it might be, there is now little doubt that epigenetic inheritance takes place. Experiments on rats have shown that female offspring who received good care when young respond better to stress in later life and themselves nurture their offspring better. Female offspring receiving low quality care nurture their offspring less well. Good maternal behaviour in rats transmits epigenetic information onto their offspring's DNA without passing through an egg or a sperm. In humans, when a mother has a condition known as gestational diabetes, the fetus is exposed to high concentrations of glucose. These high glucose concentrations cause epigenetic changes in the daughter's DNA, increasing the likelihood that she will develop gestational diabetes herself. It is thought that in sperm and eggs during the earliest stages of development, a specialized cellular mechanism searches the genome and erases its epigenetic tags in order to return the cells to a genetic clean slate. However, a few epigenetic tags escape this process and pass unchanged from parent to offspring. Epigenetics and disease. Epigenetic changes are part of normal development and health, but they can also be responsible for certain diseases. Altering any of the epigenetic processes can cause abnormal activation or silencing of genes. Such alterations have been associated with a number of diseases including cancer. In some cases, the activation of a normally inactive gene can cause cancer. In other cases, it is the inactivation of a normally active gene that gives rise to cancer. In 1983, researchers found that diseased tissue taken from patients with colorectal cancer had less DNA methylation than normal tissue from the same patients. Increased DNA methylation normally inhibits transcription or switches off genes. This means that the patients with less DNA methylation would have higher than normal gene activity. More genes were turned on. It is known that there are specific sections of DNA, ones near regions called promoter regions, that have no methylation in normal cells. However, in cancer cells, these regions become highly methylated, causing genes that should be active to switch off. This abnormality happens early in the development of cancer. We have seen that epigenetic changes do not alter the sequence of the bases in the DNA. They can, however, increase the incidence of mutations. Some active genes normally help repair DNA and so prevent cancers. In people with various types of inherited cancer, it is found that increased methylation of these genes has led to these protective genes being switched off. As a result, damaged base sequences in DNA are not repaired and so can lead to cancer. Many diseases, such as cancer, are triggered by epigenetic changes that cause certain genes to be activated or silenced. It is therefore logical to try to use these epigenetic treatments to counteract these changes. These treatments use drugs to inhibit certain enzymes involved in either histone acetylation or DNA methylation. For example, drugs that inhibit enzymes that cause DNA methylation can reactivate genes that have been silenced. Epigenetic therapy must be specifically targeted on cancer cells. If the drugs were to affect normal cells, they could affect gene transcription and make them cancerous, so causing the very disorder they're designed to cure. Another use of epigenetics in disease treatment has been the development of diagnostic tests that help detect early stages of diseases such as cancer, brain disorders, and arthritis. These tests can identify the level of DNA methylation and histone acetylation at an early stage of disease. This allows those with these diseases to seek early treatment and so have a better chance of cure. In eukaryotes and some prokaryotes, the translation of mRNA produced by a gene can be inhibited by breaking mRNA down before it is coded. Information can be translated into a polypeptide. One type of small RNA molecule that may be involved is small interfering RNA. The mechanism involving a small double-stranded section of siRNA operates as follows. An enzyme cuts large double-stranded molecules of RNA into smaller sections called small interfering RNA, siRNA. One of the two siRNA strands combines with an enzyme. The siRNA molecule guides the enzyme to a messenger RNA molecule by pairing up its bases with the complementary ones on the section of mRNA. Once in position, the enzyme cuts the mRNA into smaller sections. The mRNA is no longer capable of being translated and a polypeptide is no longer made. This means the gene has not been expressed. It has been blocked.